Dendritics team is proud to introduce its recently developed innovative tool intended for B-cell immortalization, the Blood B Booster Kit. Whatever your position as a lab worker, whether in private or academic institute, if you are interested in one of the listed fields, if your favorite species is human, mouse, rat, dog, horse, sheep or rabbit, if you are targeting normal as well as pathologic B cells for repertoire, genomic, transcriptomic or proteomic studies, thus your new tools must be the human blood B booster and the animal blood B booster kits, innovative tools intended for B-cell immortalization, developed and manufactured by Dendritics. As a brief background, it is well known that B lymphocytes are poorly represented in biological samples and that these cells have poor capacity to enter cell cycle. Thus, studying B cells generally requires an immortalization step in order to establish biological material resources. The historical method to immortalize B cells used to be the infection by the Epstein-Barr virus, but this method is poorly performant. Alternative methods currently exist to immortalize B cells, but they are associated with significant limitations. The strategy developed by Lanzavecchia's team, using CD40, TLR9 and EBV, is restricted to the memory B-cell subset. The phage display technology developed by Medarex is not only expensive and time-consuming, but also requires several rounds of screening and is limited to a single specificity. The transgenic mice technology allows only a partial transfer of human Ig genes in mice. The Ig chain's assembly occurs in a random manner, and the murine response against a human target is then elicited in a non-human context. The development of the Blood B booster kits was based both on existing approaches and on the generation of new reagents. The classical EBV infection yield was 1 immortalized cell per million. The combination of CD40 stimulation and classical EBV infection increases this number to 1% of the B cells immortalized. Dendritics has optimized the CD40 system through the replacement of irradiation by lyophilization for the transfected murine fibroblasts used in the CD40 system. In contrast to the irradiated cells, freeze-dried cells can be controlled by flow cytometry, are easily transferable, and the lyophilization process is much more reproducible than the irradiation process. Dendritics has developed a very powerful medium additive called the booster reagent, which contains more than 80 components and which increases 100-fold the CD40-induced B-cell proliferation. Dendritics has generated a novel murine monoclonal antibody, which is a pan-B. The recognized antigen is not identified yet, but this antibody increases by 20-fold the number of CD40 plus EBV immortalized B cells. And finally, with the development of 2 novel anti-EBV murine monoclonal antibodies, and also by subcloning the original EBV producing line, Dendritics has significantly improved not only the robustness of the EBV production process, but also the transformation capacity of the resulting viral suspension. The principle consists in obtaining the biological sample of interest. Human samples are processed through the human blood B booster kit, to obtain polyclonal B-cell populations representative of the global repertoire. The animal kit can be used for various species and permits the obtention of activated B cells representative of the global repertoire. These cells need to be fused with the murine sp2/0 myeloma prior to obtain immortalized B cells representative of the original repertoire. The pertinence of the donor selection and the expertise in hybridoma technology are the two keys to success that are not under the control of Dendritics technology. The human blood B booster kit uses two stimulation transformation successive steps. The first step is represented here. The freeze-dried murine fibroblast transfected with the Fc gamma receptor, the anti-CD40 antibody, the pan-B antibody, the B cells, and finally the EBV viral suspension. After 8 to 9 days of stimulation, CD40 ligand transfected freeze-dried cells are added, together with the pan-B antibody and the EBV viral suspension. All these reagents were packaged as an easy-to-use and transferable kit, which is available for any standard cell culture lab. The human blood B booster kit allows the immortalization of an average of 20% of the starting B cells, and this picture shows the chronologic evolution of a representative culture from day 0 to day 20. A magnification of a proliferating B cell is shown here, with very characteristic branched projections. The efficiency of the human blood B booster kit led us to extend this strong B cell stimulation process to several animal species. We have successfully tested mouse, rat, rabbit, sheep, horse, and dog. Splenocytes or peripheral blood cells are processed with the animal blood B booster kit, which consists of two simultaneous independent stimulations. The principle is the same than for the human kit, except the absence of EBV transformation, and except that both stimulations are run concomitantly. This strategy aimed at increasing the number of stimulated B cells. After 3 to 5 days, the cells are pooled, and then a classical fusion with the murine sp2/0 myeloma is performed. In order to establish proofs of concept, we have used the immortalized B cell lines generated through the blood B booster kits to develop human and animal monoclonal antibodies. We first established the proof of concept for the human version of the kit, and then we extended the application to animal species. The first POC is the generation of a human monoclonal antibody raised against the human platelet antigen HPA5b. The second POC was the generation of neutralizing human antibodies raised against the chikungunya virus. The extension to the animal allowed us to demonstrate that the animal kit facilitates the access to a broader repertoire in mouse, and later on we also have been able to show that our technology could be used for comparative studies between animal and human humoral responses to a given antigen. To establish the first proof of concept, we have selected a rare disease, the neonatal alloimmune thrombocytopenia, which occurs due to a mother versus fetus platelet antigen incompatibility. HPA5b is the second most frequently implicated platelet antigen in Caucasians for NAIT. Because the platelet antigen are generally expressed at very low basal level and thus are poor immunogen, And because two antigens, HPA5A and B, were very close, differing only by a single residue, no monoclonal antibody specific for HPA5B was available when these studies were started. Our objective was to demonstrate that our technology will allow the isolation of a very rare human antibody. For this purpose, we have selected a mother whose fetus had a severe NAIT, suggesting that she has developed alloimmune antibodies against her fetus. 20 ml of blood were processed through the human blood B booster kit, and we obtained the first HPA5B human monoclonal antibody available in the world. This antibody, the clone F2H11, is of IgG isotype and was tested in ELISA phenotyping test using donors which were previously genotyped by SSP-PCR. This picture shows that the F2H11 clone reveals only the HPA5B plus platelets. ELISA and SSP-PCR data were found to be correlated for 250 analyzed donors. Comparative flow cytometry studies revealed that the F2H11 clone stained the HPA5B and not the HPA5A platelets, even if these allotypes differ only by a single residue. To go further, aggregation assays were performed. Briefly, HPA5B plus platelets were induced to aggregate through incubation with collagen. The picture represents the inhibition curve of the aggregation in the presence of either the F2H11 clone or the B4512 clone, another HPA5B antibody developed during this program. We can see here that the addition of F2H11 antibody is able to reach a complete inhibition of the HPA5B plus platelets. The inhibition profile displayed by the B4F12 clone led us to extend our investigations, and interestingly, we have observed that this later clone inhibits the aggregation of the only activated platelets. The great potential of this antibody as a ReoPro-like new reagent is not discussed here but is currently under study. The second proof of concept was based on the generation of human anti-chikungunya virus monoclonal antibodies. The Chik virus is an RNA alpha virus which induces a dengue-like illness. In 2005, about 30% of the population was infected in Réunion Island, which led the French government to consider this epidemic as a priority for public health. No specific vaccine or treatment was available, leading the scientists to consider the development of antibody therapy. Our objective was to demonstrate that, using our technology, we were able to rapidly isolate, neutralizing human antibodies raised against Chik virus. We have selected a patient who has recovered from a chikungunya virus infection. 50 ml of blood were processed through the human blood B booster kit, and we obtained a large panel of anti-Chik virus human monoclonal antibodies. This first picture illustrates an immunofluorescence staining performed with a representative anti-Chik antibody. We can see that, in contrast to non-infected cells, an intense staining is obtained with chikungunya virus infected cells. On this right panel, the neutralizing capacity of the anti-Chik virus antibody is shown. Briefly, the virus is pre-incubated with either control or specific chick antibody before addition to cells for infection. This immunofluorescence staining shows that, in contrast to the control mAb, the anti-chikungunya virus antibody is able to inhibit the infection of cells. This curve shows that a concentration of 1 microgram per ml of the specific antibody is enough to totally inhibit cell infection by chikungunya virus in vitro. These first results led us to test the neutralizing capacities of our antibodies against other viruses more or less close to the chikungunya virus. This table shows various profiles with, for example, the E26D9 clone, which neutralizes the 3 different strains of chikungunya virus, as well as the Ross River and the Onion viruses. A global analysis of our data shows that the usage of the blood B booster system allows the obtention of all the immunoglobulin isotypes and can be applied to most of the antigen types. The first table shows that, depending on the targeted antigen, all the immunoglobulin isotypes could potentially be obtained. Here are listed the different types of antigens for which we have obtained human monoclonal antibodies, and it appears here that this technology could potentially impact several research areas. In an attempt to extend the process to the animal species, we first tested the animal blood B booster in mouse. Splenocytes were collected after an immunization program and were processed or not through the animal blood B booster system before the fusion with the SP20 myeloma cells. 3 independent experiments are shown here. We can see that the blood B booster kit significantly increased the percentage of Ig secreting wells from 30 to 60 to 90%. Further molecular studies demonstrated that this increase reflected a broader clonal diversity rather than a simple increased proliferation of splenocytes before fusion. Another very interesting application is the transition from the human to the animal repertoire analysis. For this demonstration, we have selected the leptospirosis model. This disease is a bacterial zoonotic disease caused by Spirochaetes leptospira. This disease affects both humans and various animal species. Our objective was to set up a model to study the humoral response to leptospira. For this purpose, we selected a human donor naturally infected by leptospira. The blood sample was processed through the human blood B booster kit, and we obtained a series of anti-leptospira human monoclonal antibodies. This picture shows an immunofluorescence staining of leptospira culture plate with some of these antibodies. We can see different profiles. Antibody 1 and 3 stain the bacteria itself. Antibody 2 stains the biofilm protecting the bacterial layer. Antibody 4 stains the cocoon-like structure around the bacteria body. These first antibodies appear to be useful tools for microbiologists to study the life cycle of leptospira. On the other side, we have collected blood sample from a dog vaccinated with a subunit of leptospira. Using the animal system followed by a fusion step, we have developed a series of canine anti-leptospira antibodies. These antibodies were tested the same way. On the right panel, we can see both similarities for the biofilm or the cocoon-like structure and discrepancies for the bacteria body when both pictures are compared. The discrepancy is consistent with the natural infection versus subunit vaccination for the human and canine donor respectively. This study illustrates that the combination of the human and the animal blood B booster systems represents a pertinent tool for vaccine design or therapy strategies in humans using animal models. The blood B booster kits are now available on the market. For pricing or ordering information, please contact your local distributor. The list of the distributor is published on the website of Dendritics. Here are some links to get technical information. Please note that Dendritics commits to provide a significant technical assistance to the users. Thank you for your attention.